All right, part 3. So we were looking at, first we have looked at purine. Now we'll look at pyrimidines. Okay, pyrimidines we have okay, so this is a pyrimidine ring. This is N1, N2, N3, 4, 5 and N6. So this N1 is what is attaching to a ribose sugar. Is what is attaching to a ribose sugar. Okay, so if the second carbon is deoxy, then it becomes deoxypyridine, pyrimidine, or if it is OH, then it becomes just pyrimidine. This is 1, this is 4, this is 5. All right, so this is C3. Now if I attach an amino sugar, if I attach an amino group here, this becomes cytosine. This becomes cytosine, ribose sugar, so it becomes cytidine, a cytosine plus a ribose sugar. This becomes cytidine and plus a ribose sugar, then it becomes cytidine. Now if I remove this oxygen, it becomes deoxyribose and this becomes deoxycytidine. So what are basically nucleosides? Nucleosides are a sugar plus our nitrogenous base. It can be either a purine or pyrimidine, a sugar plus a nitrogenous base, bond at N9 of purine and N1 of pyrimidine. All right, the sugar is ribose in a RNA and deoxyribose at second 2' carbon in DNA. So RNA and DNA. Coming to what are nucleotides? Nucleotides are nitrogen bases, that is your purines or pyrimidines, plus a sugar. Now nitrogen bases plus sugar, what does this become? Yeah, it becomes a nucleoside. So a nucleoside plus a phosphate group, a nucleoside plus a phosphate group, this is what makes it a nucleotide. Okay, now if two more phosphate groups are made, it becomes this is a mononucleotide. This is a dinucleotide and this is a trinucleotide. What is the bond formed? The first bond that is formed is an ester bond. Okay, it can either form between a 3' and a 5' hydroxyl group. Remember, this 3' and 5' is only for mononucleotides. Polynucleotides will not occur at third position. It will occur only at the fifth position. All right, it only occurs at the fifth position, not the third position. Third position is only for a mononucleotide. Okay, so the first bond between a sugar and a phosphate, either at the 3' or the 5' carbon, this is always an ester bond. It is always an ester bond. But the subsequent bonds that are formed between phosphoric acids, these are the acid anhydride bonds. These acid anhydride bonds, they have a separate pKa value wherein they act as acids, because of the high delta G. Breaking them will yield a lot of energy, minus 7 kilocalories, up to 7 kilocalories of energy. Okay, so the first bond is ester bond and the second two bonds are acid anhydride bonds. Let me show you something. Now For instance, I have a pyrimidine with me. This is the N1. This pyrimidine is attached to C1, C2, C3, C4 and C5. Okay, this is C3 and C5. So which one is most important for us? It can be either C3 or it can be either C5. Okay, so in C3 and C5, C3 and C5, you can see that C3 and C5, a phosphoric acid or phosphoric acid is attached. It can be either at a C3 or at a C5, C3 or C5. Now this bond here, what color? Okay, this bond here between the carbon and oxygen, this is the ester bond that is present. This is the ester bond that is present. Now if more phosphoric acids are attached, this is a dinucleotide and this is a trinucleotide. This is a trinucleotide. Now these bonds that are formed, this is an acid anhydride bond. These are acid anhydride bonds. Okay, suppose you attach this NH to this becomes cytidylate or it becomes a CTP. The structure here, what we are seeing is CTP. It is CTP. Now the pKa value of this is approximately 1, that is, it is very acidic, whereas this is around 6. This is around 6, and nitrogen bases, these are 10 to 11. So these are basic, but because the pKa value of this terminal phosphoric acid is so low, these act as acids. Whereas this is 6. So if you're talking about a dinucleotide like say ADP, so ADP's acidic nature or basic nature will depend upon the pH. If the pH is low, ADP will act as an acid, and if the pH is high, ADP will act as a base. So because this is very near normal, based on the pH, and dinucleotide will act, whereas a trinucleotide will always be an acid because the pKa value is near 1. All right, now so that is about it. Basic structure nucleotide. The bond is ester bond. First bond is ester bond and the other two bonds are acid anhydride bonds. Breaking this bond will yield a lot of energy, like your ATP, it breaks down to form ADP plus PI, or ATP is broken to AMP plus PPI. Both are highly exergonic reactions. They liberate energy up to 7 kilocalories or minus 35 kilojoules. Okay, so this breaking of bonds is always associated with a covalent bond formation in nucleic acids, right? Okay, so going further, isomerism, this can exist as a syn and anti-conformers. Now we know that the beta N glycosidic bond there is no freedom of rotation. No freedom of rotation. So they exist as two conformers, syn and anti. They are non-interconvertible because this beta N glycosidic bond, it cannot rotate. This cannot rotate. Like for example of syn and anti. Now let us talk about adenine. Let us say this is adenine. All right. This adenine is attached here with a ribose sugar, and this adenine is attached with a ribose sugar. Now here you can see that the ribose sugar is towards the purine. Ribose sugar is towards the purine. This is the syn form, whereas here the ribose sugar is out of the planar structure of the purine. So this is an anti form. Syn and anti form, they cannot exchange. Okay, they are not interconvertible. To form an anti from a syn, you have to break this bond. Again, you have to attach the ribose sugar to the purine. Then only it can form an anti-conformer. Both these are not interconvertible. All right. So what is the importance of this? Naturally, syn forms occur very rarely, except in zDNA or zDNA. They do not occur anywhere. Mostly it is anti form. Why? Because this will not come in between. Suppose in a DNA molecule, what is happening? They are stacking up. They are stacking up, and the sugar is out of the way. The sugar is out of the way, which is attached to a phosphate group. Sugar is out of the way, which is attached to a phosphorus group. Okay. Now what is happening here? Because the sugar is away, they are able to stack up. Suppose if that is not the case, if it is syn, what will happen is, when this is the case, the sugar will come here and it will not allow them to stack up properly. It will not allow them to stack up properly. The stacking between these two, nitrogen bases is hindered because of the presence of this sugar. That is why in naturally occurring nucleic acids, syn conformation occurs very rarely, only in zDNA or zDNA. Right? Now the peculiar properties of nitrogen bases, nucleosides are that they are aromatic. What do we mean by that? Is the ring itself is aromatic, and the covalent bonds that are present inside both purines and pyrimidines, they cause absorption of UV light at 260 nanometers at pH 7. If you increase the pH or decrease the pH, what will happen is the amount of absorption will increase or decrease. So at pH 7 we have it as 260 nanometers. Now what does this cause? One is the mutagenic effect of UV light, wherein purine and pyrimidine dimers are formed. Purine and pyrimidine dimers are formed. Like most common is thymine dimer. Okay, thymine dimer. Most common. We use UVXE nuclease or nucleoside excision repair for this. All right. Next is hyperchromaticity or hyperchromicity of denaturation. What do we mean by hyperchromicity of denaturation? DNA, it is a coil structure. Okay. Now suppose you put temperature, what will happen is it will get denatured. And it will open up. This denaturation will cause absorption to increase by 40%. To 40% increase absorption will occur, and based on this 260 nanometers we can measure the amount of nucleic acids present inside a solution. Okay, so we have seen absorption, we have seen isomerism, we have seen different bonds that are formed. All right.